I'd like to welcome everyone to this last seminar of the year. First of all, I'd like to acknowledgement of country. We acknowledge we're situated on Noongar land and that Noongar people remain the spiritual and cultural custodians of their land and continue to practice their values, languages, beliefs, and knowledge. So our first speaker will be a PhD student, Nooshin Shahbazi. So Nooshin is a student working with Ken Flower as the principal supervisor, Mike Ashworth, and myself. She's been with Ari upstairs for the last 3 odd years. And she's been looking at an innovative tool to both detect and map weeds using LiDAR. So with that, I will get Nooshin to begin her remote presentation.
Hello, everybody. Thanks, Professor Hughes, for chairing this session and introducing me. And I would also thank to my principal supervisor, Professor Flower, and my co-supervisors, Professor Becky, Professor Callow, and Dr. Ashworth from the School of Agriculture and Environment, and Professor Mian from the School of Computer Science. My work was supported by the financial support of the Australian Government Research Training Program Scholarship and the Kelowna Postgraduate Research Award 2019. I would like to thank the Australian Herbicide Resistance Initiative, ARI, which provided capital and operational funds for my project. I would also thank the Stealth Technology Company, which provided the technical support to my project. The global population has been projected to increase to more than 9 billion by 2050, which will cause to increase the demand of agricultural productivity. The production increase needs to be intensive yet environmentally friendly to secure future food supplies. However, weeds by negative impact on agricultural production can still significantly affect the crop yield, and the herbicide being the dominant way to control weeds can raise some concerns because of the inappropriate herbicide use and overuse of herbicides. Concerns such as reducing the crop yield, affecting an environment and human health, and the herbicide resistance as well, developing herbicide resistance. The increasing level of herbicide resistance have resulted in a greater focus on cultural weed control methods, such as site-specific weed management and harvest weed seed control, which they've both been very successful in the last couple of decades. However, there are some weeds, such as wild oats and brome grass, that they shed their seeds before harvest and kind of invade the effectiveness of harvest weed seed control. We also noticed that these weed species that they grow taller than the crop and they persist in the crop until harvest, they can be differentiated based on their height differences from the crop canopy. So the idea came that if we could detect these weeds and map where they were in the previous season, we could apply targeted weed management in the following year. On the other hand, by developing technology, remote sensing has become a very powerful tool in detecting weeds and mapping weeds. Remote sensing can be used for map-based and site-specific weed management approaches to collect weed data and its distribution maps in the crop for future uses. Previous studies showed that the machine learning methods such as object-based image control analysis and deep learning have been successful in detecting weeds and mapping their locations. Having this information, and we came across to a remote sensing sensor called LiDAR, which is light detection and ranging sensor. This sensor can determine the ranges by targeting the object with a laser and measuring the time for reflected light to return to the receiver. The output of LiDAR is either a 2D or a 3D point cloud of the surrounding environment. For the purpose of our project, we used a SAgE MR6000 3D LiDAR. Our LiDAR had 24 scanning layers of vertical pixels, and it had the frequency scanning was 10 Hz for this LiDAR. Our LiDAR had 120-degree horizontal field of view and 15-degree vertical field of view, and it could detect targets up to 200 meters. So we defined our research objectives, which were the main objective was we needed to understand which weeds are our targets, that we can differentiate them based on their height differences at harvest time. And also we needed to know if LiDAR can detect those weeds, and if LiDAR could detect those weeds, how accurate to detect them, measure their height, and map them in the field. And after answering all these questions, we needed to see if we can demonstrate the LiDAR use in the real world, like by putting it on a farming machinery such as harvester or a sprayer for our operations in the field. So we defined 3 research steps to answer the research questions. The first step was determining which weeds grow taller than the crop canopy. And the second step was determining if LiDAR is capable of those targets that we had in the trials, and if it was capable, how accurate it could detect the targets. And the third step was evaluating the accuracy of LiDAR to detect and geolocate the position of weeds growing above the crop canopy at harvest. So as the first step, we contacted our contacts in the Western Australia's central wheat belt. We contacted our farmers, and we identified some fields with weeds taller than the crop height at harvest time. And we conducted 2 surveys in November 2018 and November 2019. And then we studied 30 fields in 2018 and 35 in 2019. And in each field, we assessed 3 weedy patches. So we studied 191 replicants. And in each weedy patch, we recorded 30 plant height and 10 weed height of one species. The studied crops in those surveys were wheat, barley, lupin, and chickpeas. And the studied weeds were ryegrass, wild oat, wild radish, sanchez, and brome grass. We ran a sensitivity analysis by using 3 different height thresholds of the crop height, which were the mean crop height, 68 percentile of crop height, and 95 percentile of the crop height. The result of this study showed that across the 191 assessed weedy patches, in 97% of the patches with wild oat and 86% of the weedy patches with wild radish and 92% with sanchez. It was possible to distinguish the weeds taller than the 95 percentile of the crop height. So we found our targets. Our target weeds were wild oat, wild radish, and sanchez. As a second step, to answer the capability of LiDAR of detecting the targets that we were looking into, we ran 3 different trials. So the first 2 trials, we ran them with using wooden rods. And the reason of using wooden rods, we wanted to control the target's height and their width. So the first 2 trials were ran in one of the UWA lawns. So we chose rods by the height of 5 centimeter to 60 centimeter, and the width of 2 millimeter to 8 millimeter. As the first trial, we put the rods in a line with 50 centimeter spaces. All of the rods were same height and different width. LiDAR was static, and we scanned the line from the distance of 4 meters to 18 meters, and we performed 40 scans at this situation. And as the second trial, since we tend to grow in patch, we put 10 rods together with the same height and different width, and we put 4 patches, and LiDAR was looking right in the middle of the line, and we performed scans from 4 meters to 18 meters, and we performed, 4 to 20 meters, and we performed 45 scans. As the third trial, we grew a plot of wheat in Shenton Park Station in May 2019, and we also grew wheat seeds of wild oats and sanchos in pots. And in November 2019, when both weeds and wheat were mature, we transferred the pots to Shenton Park, and we located the wheat pots well above the crop height and measured the height of the weeds, and we recorded the location of the weeds, and we also measured 300 replicants of the wheat height to be able to compare at the later stage. And we put the LiDAR at a distance of 2 meters from the first row of wheat, and we performed scans. During these scans, the LiDAR was connected to an AdLink embedded computer through an Ethernet cable, and all the data was recording on that computer. However, before any recording, we could see what LiDAR can see through the software that SAgE provides with its LiDAR, and we made sure that LiDAR could see the targets before recording. The produced files from the LiDAR was a raw back file, which is robotic operation system messages recorded in a back file, and we converted that file to a point cloud with the points with the X, Y, Z of the LiDAR estimated of each point. And we could visualize those point clouds either in RVs or cloud compressed software. Then we created a moving window, and we segmented each point cloud into the target. And also we performed pre-processing steps such as removal of noise and dimension smoothing for the point cloud data. The dimensionality of each recorded point cloud was also reduced using the PCA analysis, and the data was aligned before further processes. For each trial, we performed the k-means clustering to kind of automating the target detection and assessing the accuracy of the target detection. The results showed that in the first 2 trials, the targets equal or shorter than 60 centimeters could not be detected at distances larger than 18 meters. And also the targets with the height of 5 centimeters could not be detected with the LiDAR at all. And the width of the rod, which was equal or larger than 2 millimeters, could be detected by LiDAR. And then wheat scanning showed us the 100% of the wheat targets could be detected by the LiDAR, and LiDAR could, during the scans, we could detect the LiDAR estimated height and the actual wheat height, with root mean square error of 5 to 7 centimeters. Overall, the detection of the targets by the LiDAR in these trials was the function of the LiDAR scanning range, target size, and the amount of noise that we had in those point clouds. As the third stage of the project, we grew the seeds of wild oats and sanchos in pots, and we transferred them to a wheat field in Candida, which we knew that we had natural wild oats in those fields that they grew taller than the crop. We mounted our LiDAR on a moving machine, and we, before performing scans, we recorded the location of the weeds, and we measured the height of the weeds, and we ran through the area in a rectangular shape. And after the data was collected, we performed the pre-processing programs and created a filter function in C++, and then we performed the random sample conscious plane segmentation on the point cloud, and we used 3 thresholds of mean crop height plus 2 standard deviation, mean crop height plus 3 standard deviation, and mean crop height plus 4 standard deviation as the dynamically threshold that we calculated for the estimating the inliers and outliers. By this, we mean like any, at each threshold, any inlier would be considered as the crop height, and any outlier would be considered as a weed. Then we developed our prescription map. On the prescription map, any green dots would show the known weeds that we could detect, and the blue dots shows that the weeds that we could not detect. And we create the prescription map for all 3 studied thresholds, height thresholds. And in each of the prescription maps, the white grids would show that there was no detection in them. Yellow grids, at least 1 detection were in yellow grids. Orange grids would show that the number of detection in the cell was larger than the standard deviation of the detections in that cell. And red grids showing that the number of detection in the cell was larger than 2 standard deviations. Then we calculated the accuracy of cells was calculated as a percentage of the number of correctly detected cells, and compare these percentage with the total number of cells. The study showed that up to 97% of the known weeds could be detected in this study, and this was at the lowest study threshold, which was mean plus 2 standard deviation, and we needed at least 1 detection in that cell to detect the weeds. This study demonstrated that LiDAR could be successfully used for detecting weeds with both measurement accuracy and detection accuracy. In our project, we developed the novel weed detection and mapping system of some of that we could successfully detect and map some of the common weeds in Western Australia, which was a great success for us, and which we could be using this tool that enable farmers to locate weed patches, evaluate the success of their previous weed management technologies, and also they could plan for their future strategies. And by developing this method and this application, the targeted application and the herbicides could be used in a targeted way, or we could have like non-herbicidal weed control methods. And these could reduce the input costs and the environmental impact of the herbicide. And we could reduce the amount of herbicide that we're spraying. And we can, by doing this, we can achieve the goal of like more robust increasing in agricultural production and producing more food for the growing population. However, this project is not finished yet. For the purpose of the continuing of this project, we involved a third party, and we've got a patent to commercialize this idea, but still a lot of questions are on the way to be answered. Questions such as like, when is the optimum time that LiDAR can be used? We're still thinking if we can use it on harvest, or we can do it earlier. Or questions such as, where is the optimal place on the harvester or sprayer that we can mount our LiDAR? And either we can use alternative LiDAR, we still don't know. We're still studying that part. And but the good thing is we have farmers that they're very passionate about this technology, and they're cooperating with us, and we're running our trials. They let us to go on their lands and try our application, and we're hoping that we can use this application as a service for farmers, and we can help them in reducing their input for the herbicide. Thank you so much for listening to my presentation today, and I would be happy to answer any of your questions.
Thank you, Nooshin. That was great. You provided a good overview of your research, and most importantly, you outlined some of the next steps because there's a lot of work to be done in this space. So with that, happy to take any questions from the in-person audience or from the folks online.
Thanks, Nooshin. That was a really good talk. It's Caitlin here. You showed some really nice work about how the LiDAR can be used to detect the weeds that are growing above the canopy. But I also wonder, could you comment on whether it can be used in parallel as well to also look at getting at biomass and or yield potential from the cropping systems as well as weed patches?
Understanding the, thank you for your question. Understanding the density or yield protection, or these were not the aim of our project. However, obviously, this can be a very good way for future studies. There are some studies that they've been looking for LiDAR for understanding the density, the plant density or the crop density. But in our project, it was not the aim. But the LiDAR has the capability of doing that, and we can, by using like machine learning, that is achievable.
Yeah, cool. Thank you.
You're welcome.
Any other questions? I was wondering, Nooshin, has LiDAR been used previously in other countries to detect or map weeds, or are you the first to really look at this from an agricultural perspective? I know it's been used in the mining industry, this technology, but to your knowledge, has it been used in the weed management area?
It has been used for early weed detection, and which is looking for detecting weeds in the row crops, like in the rows. But specifically, and they were mainly using like on UAVs, but specifically on the ground-based. I haven't came across any studies that they use it as a ground-based and as like an application that we can record data and map the weeds at harvest time. I haven't came across any studies.
Okay. Well, with that, thank you, Nooshin. Nooshin will be continuing to join us virtually, but we will now move to our second presenter. I'd like to present Dr. Phil Ward, who is retired from CSIRO. He's been working with Ken Flower on a crop water relations project. So I will ask our next speaker, Phil Ward, Dr. Phil Ward, to come up and give us his play.
Okay, thanks for that, Hugh. I've got to admit, when Ken first asked me to give this seminar, Ken sends his apologies, he can't be here today. When he said, Phil, can you give this seminar? My first reaction was, Ken, I'm retired. Why would I do that? It makes no sense. But then I thought very quickly, and I thought, actually, it's a really good story, what we've got here. And this has been a long-term trial. It's been a 12-year trial, and we're still finding really incredible ways of looking at the data and new ways of analysing it. And I think it's a good story, and I think it deserves to be told. So if Ken can't do it, I'm happy to help out. So what I'm talking about is what is the effect of uneven crop residue distribution as harvesters get bigger and bigger, so they're cutting more and more stuff off, and then what do you do with it when it comes out the back of the harvester? We've just heard of something you can do right in front of the harvester to map weeds. What happens to the stuff that comes out the back end of the harvester? Do you put it in a windrow? Do you try to spread it? How far can you spread it? What happens if you don't get it right? So that's the sort of thing we'll be looking at today. Thanks, obviously, to Ken for doing all the data analysis that we've had so far. Nicola's had a big role in the soil chemistry side of things. Neil accordingly did all the trial management over a very long time. And I've chipped in just to get the glory shot here for the presentation. So here's a bit of an overhead view. I'm old-fashioned, I like pointers. So here's our header moving up and down a paddock. And you can see it's spreading the residue behind, but it's not spreading evenly. There's bits here where there's no residue spread. Bits here where it's thick, bits here where it's very thin. So under the old style of driving a tractor more or less randomly around a paddock, that wasn't an issue. When you come to a controlled traffic system where you keep driving up and down the same lines all the time, you're consistently putting more or less residue in the same places all the time. What impact is that going to have? So our aim in this project is to, or in this presentation, is to understand the effects of crop rotation, so different crop species, and residue management, whether it be windrow or spread, on crop production and soil properties under a long-term no-till system. So very quickly, the key findings. Residue spread behind the harvester was uneven. And that left high in nutrients, particularly carbon and potassium, right behind the header. There was also differences. The differences in residue spread could have positive or negative effects depending on seasonal conditions. So where you had really dry conditions around sowing the next crop, residue is a good thing because that retains moisture in the system. Where you had plenty of water and frost, residue was a bad thing. And so what we want to try to do is work out a way of balancing those competing findings. So we used our long-term trial at Gunderton. Here's if you can see up there is a car. So these are big plots. These are, this is one plot, this is one plot. These are canola plot. 36 metres by 80 metres managed with commercial scale machinery. The trial went from 2007 to 2018, so a long-term 12-year trial. Controlled traffic for the entire period. I'll be showing the results from the last 6 years of that. So we've already had 6 to 12 years of consistent residue deposition, I guess, in different places. So we have two rotations I'll be comparing today. One is all cereal, which has been wheat, wheat, barley. Another diverse rotation, which is a wheat, legume, canola. Two residue management. So we've split the plots in half. In half the plot, we've done windrow burning. So we put all the residue in the windrow behind the harvester and then burnt it. And for the other half of the plot, we've spread the residue around to try to get a residue spread and an even covering. And we've looked at, right behind the harvester, 3 different locations for subplots. And I'll show you each of those in a bit more detail now. So our cereal rotation, lots of cereal stubble, wheat, wheat, barley, and that went for 12 years. Wheat, wheat, barley, all cereals for 12 years. Our diverse rotation was wheat, lupin, canola. It wasn't always lupin. It was for 3 years, it was chickpea. For 3 years, it was pea, and then lupin. So we've had a few different things there, but wheat, legume, canola. So the thing to remember here is that we're always going into a cereal residue for our cereal rotation. For the diverse rotation, this is going into a lupin stubble. So I've got different residue types going into our diverse rotation. So that's the rotations. Our residue management, we tried to spread the residue in this half plot. See our header's gone along there and we've almost got to the middle, but you can see there's definitely more residue there than there is there. And then for our windrow burning, was where the windrow was obviously until recently. And then there's not so much in between, so we've dumped all the residue in the pile here. So that's our residue management. And then as a subplot within that, we've got our 3 locations. Centre, right behind the header, mid and outer, and the same here. Centre right on the windrow, and mid and outer locations. So that's our three main treatments, or three treatments, I guess. So measurements. What I'll be presenting today is the stuff in bold. So we're looking at residue distribution. We'll be looking at the carbon and potassium in particular for soil chemistry, and we're looking at crop yield. We're writing a paper where we're putting all this other stuff in as well. So in the interest of time, I'll skip through some of it. So seasonal conditions for the 6 years that I'm presenting today. The main thing I want to point out here is in 2017, this little blue bar here is rain around seeding. So for most years, it's been pretty reasonable. For 2017, very little. So that becomes important later on. 2016, beautiful conditions, enough summer rainfall, beautiful around seeding, beautiful through the winter, and a whole lot of frost. So farmers will understand that every time you get good conditions, you get the frost. So that's also an important thing. So for 2016, frost, 2017, dry at seeding. Now, what happened when we did do our windrow? Here's a windrow before we burned it. And reasonably sensibly, looking at the residue quantity, we get 15 to 20 tonnes of our cereal in that actual metre-wide swath there. So, yeah, lots of residue. So that again in more detail. So that's what's happened straight after harvest. Probably more important is what's happened just at seeding time. So I've got the residue at seeding as well. This is obviously a very big graph, but I'll point out the important things one at a time. So here is our centre location where the windrow was. We burned it now. It was up here at 15 to 20, and now I've brought it back down to roughly the same level as where we tried to spread the residue. And that's happened. So that's a cereal residue. That's a cereal residue. For our diverse rotation, we have canola residue here. And we see where we burned, we brought that level right down. Legume residue here, where we burnt, we brought it right down. But where we tried to spread, you see what happened with the spread residue. In the centre, there's more. 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 But then I don't know why that is. That's one of those further research things, or further thought. In the centre, there's more. So we tried to spread, we haven't got it right. And that's the average over those 6 years. We can look at what's happened in a bit more detail from year to year. Again, the point to make here is that residue quantities do vary a lot from year to year, based on seasonal conditions, obviously. But where we've got our solid line here, the solid yellow line is where we've spread the residue, and that's in the centre location. That's always higher than these two red and alpha lines. And for the same with the windrow burning, the solid line, always higher than the other locations. So we have seen, so I think I've actually got a summary slide coming up now. Yes, summary so far. So yeah, we do get residue accumulated in windrows, and it is uneven. We can get up to twice as much, even when we try to spread the residue, we can get twice as much residue right behind the harvester than we get out in the wings. And if we're doing that in the same place year to year, we need to look out for that. So what happened to the soil and what happened to the subsequent crops? We'll go there now. So in the soil, first thing I'll draw your attention to is a reasonably easy thing, rotation. Nothing. The rotation by residue management, nothing significant. Rotation by location, nothing significant. Rotation by residue management by location, nothing significant, which is mildly disappointing, but does make things a whole lot easier. The only thing that came up was a very significant effect on sulphur in particular, and that's simply because we put extra sulphur on the canola crops, and we can see that in the soil chemistry. So we can explain that one pretty easily. But in terms of rotation, whether we've got a wheat, or sorry, a cereal rotation or a cereal canola legume rotation, we're getting the same sort of soil chemistry at the end. So that's pretty easy. Location behind the header, significant all the time for everything. Carbon, nitrogen, phosphorus, etc., etc. always significant. So we're seeing quite big impacts of that residue spread on the soil chemistry. So, and there's a couple of interactions that are also significant. So residue management by location. And I'll look at those in a bit more detail now. So first of all, for carbon, where we've, the obvious one, where we windrow burn, we end up with more carbon in the windrow than we do outside of it. And that's a bit counterintuitive. We've just burned this windrow. Now we've got carbon there. What I suspect is happening here is we've lost a lot of carbon as gas, but the stuff that's left behind is black charcoal, and that's pretty resistant to further degradation. So we're seeing higher carbon where we've windrow burned. For the spread, we've got higher carbon right behind the header, which you'd expect, lower in the mid, and again, higher out here. What's going on there? More research required, more thought necessary. It is possible that in some years, our header was actually throwing straw too far, and we were getting a double overlap there. So that might be a possibility. We need to look at our numbers to see if that's what's going on there. For the potassium, we was trying to spread the residue. Not much of an effect there, which is good. So that's why we're getting this interaction. No effect for spread. For windrow burn, potassium in the windrow and not in other places. And this is reasonably well known, at least anecdotally. Farmers say, well, we've got a windrow, but we burned it. Potassium's through the roof after that. So this is the first data I've seen on it. So that's good to know. But it's backing up things that we have seen. So this is a reasonably expected result. These numbers for this particular soil type, potassium's not a problem. So we don't need to worry about it. In a sandier soil type, that's going to become a real issue that needs managing. So what happens at crop yield? Here's a very big table, one of Ken's specialties. A lot going on there. So to make it easier, in the diverse rotation, lupin yield and canola yield didn't vary a whole lot depending on residue management or location. So we don't need to worry too much about them. I'm not sure why that would be, but we do see these other impacts here where barley and wheat in the cereal rotation were affected. Now sometimes with the green ones, it's a positive impact. So there and there, more residue has given us higher yield. That's great, we love that. There are times where more residue has given us lower yield. That's not so good. We need to know what's going on there and how to manage it. And there are times where we've seen differences, and they are related to our treatments of location and residue management, but they're not actually related to residue quantity. So we need to understand what's going on there as well. And I'll show you just a few examples of each of those. So first of all, where have we had a positive impact? So here's our residue effect on barley yield, 2017. You take that point out, which is a little bit of an outlet. It's a pretty solid relationship between crop yield and residue quantity. So that's nice. And what's going on here in 2017? 2017 is this year where we've got almost no rain around seeding. So in this situation, we've got lots of residue, probably impacting on soil water. We need to look in that a bit more, but it does make sense. Lots of residue giving us higher yields with more yield with more residue under those really dry conditions at seeding. So come back to this negative effect now. That's a little disappointing. We are going up to pretty high residue loads here, obviously, 10 to 12 tonnes. That's a lot of straw on the surface. But even when we've got lower numbers, a very strong correlation, more residue giving us less yield. And this is 2016. Really good growing conditions. So we don't need our residue to protect water. There's plenty of water there. And that's an ounce of frost. And we do also know that residue can impact on how much radiation gets reflected back up into the air. So it can exacerbate frost. So where we've had bad frost, yeah, more residue is giving us lower yields. Not really the story we want to get across, but it is what it is. Interestingly, where we had canola residue instead of cereal residue, we're not getting up to 10 tonnes anymore, we're only getting up to 6, but that's a flat line. So in the canola system, in the diverse rotation, there's no negative impact of residue on our crop yields. So that's a good story. And now I'll look at one of our indirect effects. So we have a significant interaction here of in our diverse rotation. So here's what's happened. There is a significant effect, but it's not to do with residue quantity. So I plot it all against residue quantity, and that's a straight line. There's no relationship there at all. What we can see on a bit of a closer inspection is that these circles, which are the mid and outer locations, tend to be yielding lower than the squares, which are the centre locations. So I can put that in a nice little column chart like that. So in the centre locations, we're getting higher yield than the mid and outer. And on this occasion, it's not related to residue quantity. So what's going on? Well, we know that in that centre location, we've got higher nitrogen, potassium, phosphorus, sulphur, etc., etc., etc. So it's quite likely that this is a crop nutrition phenomenon going on. So our key findings. Residue spread is uneven behind the header. No matter how much we try to make it even, it is uneven. Where we're doing that with controlled traffic and doing it consistently from year to year, we need to be able to manage that. We get higher nutrition, better nutrition, right behind the header where there's most of the residue going. And that makes sense. If you're doing the same thing year after year, you're going to be accumulating nutrients there. The cereals produce more residue than the legumes and the canola. Again, that's not a new finding, but in terms of how we put that into a system, that becomes important, because it's the cereal residues, and they build up to big numbers, that were giving us issues in terms of, in frost, we were seeing negative effects. So we need to be able to manage around that. Under really dry conditions, more residue is a good thing, so we like that. But under, where we don't need to protect the water, where we do need to protect against frost, residue is not such a good thing. Okay. So the implications for a broader system, we really need to pay a lot more attention to the spreading or the residue handling behind our harvester. Is it spread? If we're trying to spread it, how accurate is it? If we're trying to windrow groom it, what effect is that having if we're doing it in the same place year after year? And the positive effects of high residue and the negative effects of high residue, we can minimise the negative effects, maximise the chance of positive effects by keeping our cereal residue under control. We don't need to get much higher than 6 tonnes, really. And we can achieve that by going to a more diverse rotation and not having cereal all the time. So that's where we are. Thank you very much.
Thank you, Phil. Do you have any burning questions from the audience?
Oh, boom, boom.
I was going to ask you, Phil, in grower paddocks, I know this is a long-term trial. Do you think your residue spread is what you would see out in a typical grower paddocks with their big headers and so forth?
Yeah, it's a good question. Yes, I do. We were using farm-scale machinery for this trial, so that was one of the reasons we did the trial the way we did. But one of the things I didn't mention was that because this was being managed by 𝒈𝒓, they would use the machinery that was available at the time. So up until the last 2 years, we're using a case harvester, and the last 2 years, we're using a John Deere harvester. Different harvesters, different residue spreads. Right. And so, all harvesters are not equal. Yeah. So I think you really need to be aware of what residue spread you're actually getting. That's the bottom line there. Yeah.
Stop sharing it will show up.
One other question I had, Phil.
Stop sharing.
You talk about crop yield. Did you measure crop emergence? Was there any did emergence affect yield?
We did measure crop emergence. As you'd expect, there's a pretty decent correlation between emergence and yield. Yeah. I haven't got that data with me just at the moment, unfortunately. Yeah. They will be part of the paper, so you need to read the paper when it comes out.
Good plug.
Yeah. So,
yes. Thank you. That was a really excellent talk. And it's a beautiful long-term data set. I really love seeing those long-term data sets. And I'm curious about if you notice, this might be a bit of an aside, but if you notice differences in, particularly in soil nutrients, carbon, because I'm very interested in carbon, at deeper levels below the system, because you had your very much your cereal rotation, but I'm interested in particularly if there was a difference between cereals and the diverse rotations, particularly through the profile if you've looked at those data.
Yeah, that was one of the things we were interested in looking at as well. So, yeah, good question again. No, unfortunately, we only measured 0 to 30. We did in 10 centimetre increments. We didn't go down below that. So maybe there's something happening down there, I don't know. But in that 0 to 30, we were hoping there'd be differences in carbon storage, but we couldn't pick anything up even over that 12-year period, except for the stuff we've put up here. So based on location, yeah, we can see the differences, but based on rotation, no.
I really enjoyed your presentation. Thank you. I imagine you would have wanted to keep your trials very clean, and they would have been. Can you make any sort of comment on what would happen with like disease susceptibility with different levels of stubble residue and soil microbes?
Can't tell you much about the microbes. We never got enough money to do that in any real sense. What was the other part? Disease, yeah. Yeah, we did do Ken wrote a really good paper about the progression of diseases and nematodes based on the different rotations. And yeah, we did see some quite big differences between the rotations. In terms of residue, there wasn't a whole lot of stuff that we saw residue. We couldn't see any evidence of residue bringing forward those cereal diseases that you'd think it probably would. So that was encouraging. But we did see more disease in that in the cereal rotation than we saw in the diverse rotation. So, again, that's not rocket science, but it's nice to be able to show that.
So, Phil, is that experiment finished now? Or? Okay, yeah.
Yep, there. So, yeah, it ran from 2012 up to 2018. We ran wheat over the entire area of 2019 as a sort of balancing thing at the end. As far as I know, we haven't been there since. I haven't been there since.
Okay, well, thank you, Phil. Let's give Phil another round of applause for that presentation. Thank you all for joining us in person and online. And as I said, this is the last seminar of the year, so enjoy your holiday break, and we'll see you in 2022. Thank you.